Hello everyone, I'd like to talk with you about carbohydrate, lipid, and protein metabolism. We'll start with the carbohydrates. So remember that metabolism is the sum total of all the chemical reactions in your body. So this includes building up new molecules that are more complex, and it includes breaking down those more complex molecules into simpler molecules. So glycogenesis, you can see the word glycogen in there, also genesis. So this is a creation of glycogen. Genesis is creation, glyco referring to glycogen. This is the polysaccharide that's the only stored carbohydrate in humans. So glycogen is how you'll store some of your glucose. Insulin is a hormone that will be important in this process because it stimulates the hepatocytes, the cells in your liver, and also your skeletal muscle cells to create this glycogen from glucose. You can also break that glycogen apart, glycogenolysis, and this is where you break down the glycogen that you've stored in your hepatocytes. And you break that down back into the glucose monomers, and now that glucose is ready for your blood. So you can build up glycogen with glycogenesis, and you can break it down with glycogenolysis. You can also create glucose from non-carbohydrate sources, and this is gluconeogenesis. So gluco for glucose, neo for new, genesis is creation. So the creation of new glucose from non-carbohydrate sources. And so this is the creation of glucose from proteins, from lipids. You can take glycerol, which is part of triglycerides. You can take lactic acid. Even some of your amino acids can be converted to glucose. If you have cortisol being secreted, which is your stress hormone, you want to put glucose in your blood. Glucagon is another hormone that is involved with increasing your blood glucose level. These will stimulate gluconeogenesis. So you can take lactic acid or certain amino acids. They can be converted to pyruvic acid, which you may remember is the final step in glycolysis. And then basically you can go backwards through glycolysis to end back up with glucose. Glycerol is also able to kind of hop into this glycolysis pathway and move backwards to your initial starting step of glycolysis, which is glucose. Lipid metabolism has to be coupled with protein. And the reason for this is because lipids are not very soluble in your watery blood. And so to help them be transported in the blood and even to be transported in your small intestine to the absorptive cells that will eventually get them to a lacteal and then the blood, lipids are coupled with proteins. And there are particular proteins in the outer shell called apoproteins. And these have specific functions, and they function as transport vesicles. So it helps make sure that those lipids get where they need to go. So by adding proteins to your lipids, you help make them a little bit more water soluble and easier to transport. And when you add these proteins to your lipid, they are called lipoproteins. And there are a couple of different types of lipoproteins. You have chylomicrons. These are what form in the small intestine in the mucosal epithelial cells. And so when you're in the absorptive cell and trying to move your lipids through the absorptive cell, this is where you're going to add the protein coat. And then these will, from here, move into a lacteal. So chylomicrons are kind of the very first step as you're absorbing those lipids. Very low density lipoproteins, or VLDLs, these are created in hepatocytes, so the cells in the liver, and they transport lipids from the liver to adipose tissue. So it helps put them into storage, your lipids into storage. Eventually, your VLDLs will become LDLs, which are low density lipoproteins. And this is when triglycerides have been removed by your adipose cells. And then we have low density lipoproteins, LDLs, and high density lipoproteins, HDLs. And these are kind of nicknamed your good and bad cholesterol, even though there's still a lot more than just cholesterol that's in here. You have the lipids, you've got triglyceride, you've got proteins, but when you're looking at the LDLs, the reason that they're called bad cholesterol, that's their nickname, is because they carry 75% of the total cholesterol in your blood, which is quite a lot. Also, there's a difference in how these different lipoproteins, the direction in which they transport the cholesterol. So LDLs deliver cholesterol to your body cells for repair and synthesis. And cholesterol is not totally bad. If you have excess cholesterol, that's bad. But cholesterol is an important component of your cell membranes, for example, in the creation of some hormones. So some cholesterol is important to have. But if you have excess cholesterol, then these LDLs can deposit cholesterol in fatty plaques along the insides of your blood vessels, and that can be bad. Contributes to atherosclerosis, higher blood pressure, and all of those problems. HDLs, your high-density lipoproteins, are nicknamed your good cholesterol. These have a lot more phospholipids and a lot more protein and less cholesterol. In fact, they remove cholesterol from your body cells as well as your blood and takes it to the liver for elimination in the bile. So LDLs carry cholesterol to your body cells, and it can deposit that cholesterol in fatty plaques. HDLs are going the other way. They take away cholesterol from your body cells and your blood, and they help take it to your liver so that you can get rid of your excess cholesterol. So HDLs are a good thing to have, and LDLs, you don't want too many of those. Here's a picture of what those lipoproteins look like, and this shows kind of the difference in the proportion of protein and cholesterol and phospholipid. So here's your chylomicron. You can see it's a little bit of phospholipid. You can see the cholesterol. You have just a little bit on the inside, and then here's your protein, so not too much. And then here is your VLDL, very low density hyperprotein, has a little bit more triglyceride, has a pretty good amount of cholesterol, and here are your proteins. Here are your low density lipoproteins and high density lipoproteins. Look at the difference in the cholesterol. Much less cholesterol in your HDL, a lot more cholesterol in your LDL. And then here is the pathway we discussed. So chylomicron pathway, as you're absorbing those lipids, they're packaged up into chylomicrons in the absorptive cells of the small intestine. And then from there, your chylomicron, so the protein with the lipid, will go into the lymphatic capillary or a lacteal, and then that lymph is eventually returned to the bloodstream through the subclavian vein. After you return those chylomicrons eventually to the bloodstream, there is an enzyme that lines the inside of your endothelial cells called lipoprotein lipase. And this enzyme helps you to remove the triglycerides out of the chylomicron. So you get them out of that protein coat. And then those lipids are ready for storage in adipocytes or in other cells. For your other lipoproteins, so the remnants that are left over of your chylomicron, those go to the liver so that you can dispose of those. Your liver creates VLDLs, which are your very low density lipoproteins. Your triglycerides are removed and stored in adipocytes. And then all of your VLDLs eventually will become LDLs. So instead of very low density, just regular low density lipoproteins. And these mainly contain cholesterol. You already got rid of the triglycerides, right? So now you have mainly cholesterol, and those LDLs get absorbed by your body cells through receptor-mediated endocytosis, which basically means you have a receptor that is able to recognize and bind to these LDLs and engulf them into the cell. And so now you have all this cholesterol in the cell. And along the way, it is possible for some of this cholesterol to become deposited inside your blood vessels, possibly forming plaques. So that's the VLDL and LDL pathway. Your liver is also responsible for producing empty HDL shells. So high density lipoprotein, remember this one is your good cholesterol. shells will pick up cholesterol from your tissues, from your body cells, from inside of your blood vessels, and you can return those filled up LDLs back to the liver. The liver then takes that excess cholesterol and the bile acids, and you end up excreting that through your digestive tract. And so HDL is your good cholesterol that helps to remove that excess cholesterol and get it out of your body. As we discussed before, cholesterol is not bad, it's necessary, but it's so easy to consume more cholesterol than you need. And your liver can produce cholesterol, so you really don't need many dietary sources of cholesterol. So most of it's actually synthesized by your hepatocytes. If you eat fatty foods, this can increase your cholesterol. And so especially you would like to avoid foods that are made from saturated fat. It also stimulates the reabsorption of cholesterol-containing bile. And so fatty foods, you want to try to stay away from eating too many of those. As your cholesterol goes up, so does your risk of coronary artery disease. Like we talked about, you can have that cholesterol that is deposited on the inside of your blood vessels thanks to your LDLs. If you have cholesterol that becomes elevated, diet, staying away from saturated fats, exercise, and there are certain cholesterol-reducing drugs that can help. Cholesterol is needed to form bile salts, to form steroid hormones, to form vitamin D, and it's an important component of your plasma membranes. So you do need some cholesterol. When you are using lipids either for storage or breaking them down, you can break down lipids and oxidize them to make ATP for your cells. And if you're not using it, then you can store that in adipose tissue. The main function of your adipose tissue is to take your triglycerides out of your chylomicrons and your VLDLs, and then you store it until it's needed. And while you do have other energy storage sources, 98% of all of your body energy reserves is in your adipose tissue. You're storing triglycerides. When those chylomicrons are moving through the blood, you have an enzyme called lipoprotein lipase that can help remove those triglycerides. And then from there, the fatty acids will diffuse into the hepatocytes, your liver cells, and your adipose cells, your fat tissue, and then it reforms into triglycerides. Your lipids have an amazing ability to be broken down and reform again. Triglycerides get broken down into glycerol and fatty acids, monoglycerides, and then it's very easy for them to reform into triglycerides. So lipolysis is where you are breaking down lipids. So lipo for lipids, lysis means to break. So the catabolic process where you're breaking apart lipids is called lipolysis. And you break off the glycerol and you break off those fatty acid chains. This needs to be done in the muscle, liver, and adipose tissue to oxidize your fatty acids in order to create ATP. If you have epinephrine or norepinephrine that's being released, those hormones, neurotransmitters, will enhance this process. If you need to create lipids for storage, that's called lipogenesis. So genesis is creation, lipo referring to lipids, so creation of lipids, lipogenesis. And this is an anabolic process, and so you're synthesizing a larger, more complex molecule. And this can be done by your liver cells and adipocytes. And you can create lipids from fatty acids and glycerol, but also from glucose or amino acids. So if you have excess sugar in your blood, then ultimately it will be converted into lipids for long-term storage. And so this is what's happening if you consume more calories than your body needs for ATP production. All of the rest goes into storage. Proteins can also be oxidized to create ATP, or you can put them together to create new proteins. Excess dietary amino acids are not excreted. Same idea with glucose or lipids. Your body never gets rid of any energy molecules that it potentially could use for ATP. So if you have excess amino acids, you can convert them to glucose, so that's gluconeogenesis, Or you can also convert proteins to triglycerides. So your body is very good at converting different energy molecules into whatever form is most efficient. If you break apart your proteins, like if you have an old worn out protein that needs to be recycled, you can deaminate, remove the amino group from that amino acid, and then you can send that amino acid into the Krebs cycle or the citric acid cycle. But this produces ammonia, which is toxic. And so your liver cells, your hepatocytes, will convert it to urea, and then that urea is excreted in your urine. Urea is a much safer form to excrete that ammonia. Your body generally prefers to use carbohydrate sources first, and because they're the easiest to use, and then they'll move to triglycerides, to lipids. Generally, your body saves the protein because proteins are used to build so many different things in your body. You don't want to be breaking down your cells. You don't want to be breaking down muscle. But if needed, you can start breaking down different proteins that are in your body. Protein anabolism is when you build proteins, and there's 10 essential amino acids in humans. And you have to make sure that you consume all of these in the diet because you cannot synthesize them. So if you eat foods that are complete proteins, such as fish, beef, poultry, eggs, these will provide all of the amino acids that you need. If you are vegetarian or vegan, that's no problem. You can consume multiple types of incomplete proteins to make sure that you get all of your essential amino acids.